internet connection. But
yeah, so you're live and you're on YouTube. So let me just briefly introduce you, Peter, so we can get this started, right? So I'm glad to be introducing today Peter Hinow, who is a professor in mathematics at the University of Wisconsin-Milwaukee. Peter is a specialist in partial differential equations and its applications to mathematical biology. He has extensive experience with lots of different mathematical biology problems, including marine biology, from what I see in your published literature, but also in mathematical oncology, which I think is what the subject for today is going to be. So you did your degree in maths at the Technical University of Dresden, where I spent a few years myself as well. Did your PhD in Vanderbilt University under Glenn Webb, so one of the legends of mathematical biology. Went on to do a postdoc at Minnesota Institute of Mathematical Applications, and then started his tenure track position at Milwaukee. You went through the ranks, and now you're a full professor. So we're looking forward to see your presentation, Peter. The stage is yours.
Thank you very much, David, for this kind introduction. And yeah, so first of all, I want to say just a little bit about what I do. So I trained in differential equations and dynamical systems, and then I found that it's much fun to apply those things to problems in life sciences in the widest possible sense. And so I've worked with pharmaceutical scientists at the University of Otago in New Zealand. That's ongoing work on mathematical modeling of drug delivery to the brain, where you have to cross the blood-brain barrier. And so the idea that they have put forward is to use ultrasound sensitive liposomes, which are small particles which contain a drug, and that makes it possible to release them in exactly the location where you want them to be. And yeah, as David mentioned, so I've also found really fun in looking at small crustaceans, so just about 1 millimeter in size, and they have an enormously complex life. They need to find food, they need to mate, they live in darkness, so they have really fine senses, and they're exploring their marine environment. And then also a recent project was to find electrophysiological recordings from somewhat artificial cardiomyocytes, and that was with a PhD student of mine. So what I want to talk about today is joint work with Emily Wang and Gabriela Pinto. Emily is a cancer biologist, and I have known her since my days at Vanderbilt, where she was a postdoc at the time. And a couple of years ago, I visited Ohio State University, and in the coffee room, I bumped into Kasia. Is Kasia in the room? And she told me she just recently visited Emily at UC San Diego, and that gave me the idea to send her an email and to ask, do you have something to work on? And this paper is basically the outgrowth of meeting Kasia in the coffee room at MBI. So this goes to show how important it is to actually travel and to meet people and to communicate. And Gabriela is a colleague of mine in our department. So I first give you a brief overview of what's going to happen. So I'm going to say something about cancer as an ecosystem, which is a not that new anymore view. And in particular, we're going to look at cancer-associated fibroblasts. And these cancer-associated fibroblasts are involved in a process that is able to turn ammonium into glutamine. And that is expected to support the cancer cells with which they coexist in a tumor. And so I'll show you some experiments that were done in Emily's lab, and then our mathematical model and how it looks compared to the experimental data, and what is worth for future considerations. So it's well known that tumors consist of not just cancer cells, they can also contain endothelial cells, immune cells, and fibroblasts, which all maintain the original genome of the organism. And we want to focus in particular on these cancer-associated fibroblasts. They have their origin in the normal fibroblasts in the tissue, but they can have different properties than healthy fibroblasts. And here's a picture of a mammary gland in the normal state, and where there is just a little bit connective tissue, whereas in a carcinoma, that connective tissue grows rapidly and basically takes over the location. And yeah, so as I said, they are involved in the glutamine ammonium metabolism. So glutamine is an amino acid, and it is needed by all types of cells for protein synthesis. And because cancer cells are rapidly growing, they need especially large amounts of glutamines. And in the process of protein synthesis arises the ammonium, which is a toxic waste product. And normally it would be removed by blood vessels and then later on excreted through the urinary system. But prior to the tumor being vascularized, the tumor has no access to blood vessels. And so the ammonium would accumulate in the vicinity of the tumor. And that causes stress to the tumor cells. Now it has been found, it was discovered a few years ago that cancer cells can recycle ammonium to glutamate. But when they do that, they have to pay a price for that, which is the decline of their number. And so that's the one of the outcomes of this work. And yeah, so here's a picture. The cancer cells, they are able to reprogram normal fibroblasts into cancer-associated fibroblasts, so that we will denote as CAFs. And actually, Emily's work in her lab is mainly focused on this arrow, on this process by which the cancer cells turn fibroblasts into cancer-associated fibroblasts. And they have discovered which miRNA plays a role, and this is excreted in extracellular vesicles, and so on. In the experiments that I'm going to talk about today, they have made two types of cultures. One, a monoculture of either cancer cells or cancer-associated fibroblasts, and another one, a co-culture of the two cell types. And here's a schematic model. So the cancer cells, they convert glutamine into ammonia when glutamine is present. And also they are able to convert ammonia into glutamine when ammonia is present. Whereas the cancer-associated fibroblasts, they do that too. But in a co-culture, it is expected that the cancer-associated fibroblasts support the cancer cell population. So they are able to help the cancer through a period of high ammonia concentration. And we want to combine experiments and mathematical modeling to get a better understanding of this interaction between the cancer cells and the cancer-associated fibroblasts. As far as the exchange of glutamine and ammonia is concerned. So here is a brief description of the experiments. They were done by Wei Yan, who was at the time a postdoc in Emily's lab at UC San Diego. So what they did, they used a certain type of breast cancer cell line in a monoculture, and also patient-derived primary fibroblasts pre-treated with extracellular vesicles collected from these cancer cells. So they have a protocol which allows them to say that those fibroblasts are then cancer-associated fibroblasts. And so those two cell types, they were grown in monoculture, and then they also made a 50-50 co-culture of both. And they measured both the glutamine and the ammonium concentration and the live cell number over a course of 3 days. And all the experiments were done in triplicates. And what's different in the different individual experiments is the initial concentration of either ammonium or glutamine. So they made some experiments where they gave glutamine at different concentrations, and some experiments where they gave ammonium at different initial concentration. Yeah, and so we constructed the mathematical model.
Can I ask a quick question about that? If you just go back a slide, the fibroblasts there, you're saying they're pre-treated with the extracellular vesicles from tumor cells. Is the idea that's going to transform them into cancer-associated fibroblasts?
That's right. That's exactly right. Yes.
So, and that's a known way to do it.
Yes, I guess you would have to ask Emily about that.
Right.
But yeah, so that's I think a pretty standard procedure in their lab.
Right. It's just the fact that it's particularly extracellular vesicles. Right, you know, why I've seen people use media or you know, coculture or whatever, but it's the vesicles specifically.
Yeah, I actually, that's out of my expertise, but I do think that I mean, their primary research effort is these extracellular vesicles and what they contain, and yeah, what that does to the fibroblasts. Yeah. So yeah, we when they showed me this, and I thought, well, I can write ODEs on that, right? So there are several processes going on. So there's the conversion of glutamine to ammonia, and vice versa, and then the cells, they grow on glutamine, and they have a death term that's caused by the ammonia. And then to make the matter work, we do need an additional unspecified source of chemical energy to make this ammonia to glutamine conversion possible. Right? If you see that, you know, they can live on glutamine and turn it into ammonia, but then they can also turn the ammonia back into glutamine. If you didn't have anything else, then that would be something like a chemical perpetuum mobile. And so we need some other source which makes this conversion of ammonia back to glutamine possible. And yeah, so here is the system of ordinary differential equation, which is the monoculture model for the cancer-associated fibroblast. So we have equations for glutamine, ammonia, which is a waste product, then this background energy source, and then the cell number. And I'll walk you through these terms and what each and every one of them means. And yeah, so first we have some sort of a balance equation for the concentrations of glutamine and ammonia. So the glutamine is being consumed by the presence of the calves, and it's turned into ammonia. And then conversely, the calves can also turn ammonia back into glutamine, but for this they need the presence of this energy that we denoted by R here. And then there is a background ammonia production that's independent of these two processes. And then we have this consumption or depletion of the chemical energy due to the conversion of ammonia into glutamine. And we have another background consumption of this chemical energy. So I should say that in the experiments, they used some nutrient medium. And if I remember correctly, what we think is this energy source is alpha ketoglutarate. But it was not measured. So this is something like a hidden variable. And then the calves, they grow on that, on the presence of glutamine in this nonlinear fashion. So we have a growth term which saturates at high concentration. So the idea is that if the concentration of glutamine is very high, it does not translate into further growth of the calves. That's why we have this rational function here. And then they also have a ability to grow on this background energy source. And then we have two death terms, which one is related to the presence of the ammonia, and another one is simply a background cell death rate. So everything was grown in Petri dishes, and so they have a limited lifespan if you can keep them alive for 3 days without repleting the medium. That's already pretty good. Yeah, so a couple of so everything that's a capital letter is a variable. Everything that's a lowercase letter is a parameter, with the exception of these capital K1 and capital K2. And just a few comments on some of them. So here we have these this minus plus plus minus matrix, right? Which in an ideal world would have exactly the same top row as the bottom row, just with the signs reversed, right? However, it turns out that we need some dimensionless number to account for how much ammonia is actually produced from glutamine, and conversely, how much glutamine is actually produced from ammonia. And this is not dictated by the chemistry, so it's not the number of nitrogen atoms or something like this. It will turn out that these are non-integer numbers. And yeah, so because we couldn't find the parameters in the literature, so we simply resorted to fitting the model output to the experimental data using standard minimization procedure, Nelder-Mead algorithm. And so let's take a look at the data and the output.
Can I ask a question real quick?
Yes, yes.
So can you say how many parameters and variables are you looking at?
Very good question. Yes, I'll have a table. I think it's
okay. All right.
10 for the monoculture model.
Okay.
We'll come back to that in a moment. Yes.
Okay.
Yeah, it's neither too many nor too few, I would say. Yeah.
Okay. Thank you.
So here are some. Here's a look at the data that we got and the fitted output of the model. And yeah, so they this is one set of data where they supplied the cancer-associated fibroblasts with varying concentrations of ammonia, so 5, 10, and 25 millimolar. And they did that in triplicates, and here you can see that they are reasonably close to each other. So they did it 3 times, but it's actually quite amazing how close those symbols are in those replicates. So they started at 5, 10, and 25. And so you see that the ammonia goes down and the glutamine goes up over a period of 72 hours. And the initial number of cells was 80,000. And so it grows over that period of time. So this is amazing. The calves are able to grow in the presence of ammonia, at least at low to moderate concentrations of ammonia. And only at the highest concentration of ammonia, they grow initially a little bit and then they decline in their number. And I should say this is the number of live calves. They had also dead cells, but that they then swim on the top of the medium, so they only counted the live calves. And
is there a reason why in the 25 millimolar there's less initial cells?
No, it's all 80,000.
Oh, is it? It's just the axis is different.
Yes.
Yeah. They don't really grow or decay. They just kind of are flat, like, you know.
Yeah. So I mean, initially they so this is after 24 hours. It's a little bit bigger, but then it goes down. Yes.
But if you put that on the same axis as your 150,000, it looked like a straight line, right?
Yeah. It would probably look like this. Yeah. So, and then they also made the monoculture experiment with supply of glutamine and at 1, 2, and 4 millimolar. And there you see that the glutamine is consumed and it's converted into ammonia. And then you have growth of the total cell number. And so you can already start to see there are a couple of cases where you have a discrepancy of the fitted model from the experimental data. I just wanted to say that the whole data set, so all monoculture experiments were used to fit just the parameters. And here is the answer to your question. This is the table of the parameter values for the calves together with the 95% confidence interval. And this was done by Gabriela. So she used a bootstrapping method to obtain these 95% confidence intervals. And so the algorithm is the classical fitting of this squared error to the experimental data using Nelder-Mead algorithm. So I can't really comment on any one of these values. I mean, we have to live with the fact that these have not been reported anywhere at all. I'll come later on towards the end to some comparison with other cell types and cell lines for the cancer cells. But for the cancer-associated fibroblasts, to the best of my knowledge, nobody has ever done such experiments and reported any such parameter values. So then we switch over to the cancer cells, and the model is essentially the same. I'm using a different letter here. So X was cancer-associated fibroblasts. Y now stands for the number of cancer cells. There's one parameter which we intentionally set equal to 0, which was the rate of growth on the background energy source. So otherwise, the model is exactly the same, but of course, we're going to obtain different parameters. And so here we have the cancer cell monoculture supplied with ammonia, again at 5, 10, and 25 millimolar over a period of 72 hours. And again, we see the same, roughly the same picture. The ammonia goes down, the glutamine goes up, but at the same time, also the number of the cells decreases considerably. So it goes down to nearly 2/3 or even a half at the higher concentration. And then if the cancer cells are supplied with glutamine, then of course they thrive. They convert the glutamine into ammonia, and their number increases and can increase quite fast. Yeah, so, and again, this set of data was used to obtain the parameters by fitting the model to the experimental results. And again, you can say what is happening here, what is happening at some intermediate points, but by and large, we're reasonably satisfied with this quality. And here is the corresponding list of parameters for the cancer cells, again with the 95% confidence interval. So one of them, two of the parameters from the previous table are missing. There is no R2 here.
Well, I have more questions. So these look to me, from the 95% confidence intervals, it looks like the parameters seem to be pretty precisely estimated. But then I'm, going back to what you commented on some of those graphs in the previous slide, especially on the left when you're giving ammonium, looks like you're really, the model is really underestimating these dynamics for glutamine and ammonium. Like, does that concern you?
Well, yeah. I mean, looking at the entire picture, I think the fit is reasonably good, right? So to improve it would require to make the model more complex, right? And with more parameters, you have more room to fit, and of course, it will get better, right?
Right.
And so that brings you to the question: what amount of complexity? Right. What amount of complexity is required to explain the experimental data at hand, and how do you justify introducing more and more terms, right? Actually, we will have. I will come back to that question when we look at the coculture data. Yes, and
okay. And one real quick. Last thing, have you looked at, okay, well, what was it, correlations between parameters? Sorry, you're doing sensitivity analysis.
Yes.
Sorry, I'll be quiet.
Right.
Go on. Sorry.
Yeah, that is a good point. So when you normalize all these parameters to value 1, and then you plot the 95% confidence intervals, that gives you an idea on their relative sensitivity, right? So here is a plot of these parameters with their confidence intervals, and when you have such a very narrow interval, then that suggests that this is a highly sensitive parameter, and it would be advisable to devote more attention to that and to see if you can determine it in other ways. And so one that is quite common to both is this R1, which is the rate of growth of cells on the glutamine. Right, so this is sensitive for both populations of cells. Yeah, and then to obtain the model for the coculture, we just merged the two models for the cancer cells and the cancer associated fibroblasts. So it's really putting the two of them together. And so now every parameter has this superscript, either CC or CAF, right? So those are the ones that were obtained already from the previous set of experiments. And now we're trying to use those same parameters to explain the results from the 50-50 coculture. So there are no additional cross interaction terms, but I'll have to come back to that again at a later stage. So actually, we wanted to keep the number of changes relative to the monoculture small, and it turns out that we can get away with just 2 adjustments by a small amount. So we have to change the growth rates of the calves and of the cancer cells by just a little bit. Once we divide by 10, the other one, it's next to nothing. So we multiply it by 1.08. So one disclaimer, this is not intended to say these are unique and no other values would do the same result. But at the very least, we're not forced to redo the entire fitting process when we are confronted with the coculture data. So here is the result of that model acting on the coculture data. So the cell number is now the number of all kinds of cells, but we'll also have the fraction of the cancer cells that will come in the next slide. So here, when we supply this population with ammonia, then at 5, 10, and 25 millimolar, so we see that the ammonia goes down and the glutamine goes up, and the total population, well, it increases and then it decreases. And we're not really good at achieving that with our model, and we'll have to come back to that at a later stage. So the percentage of live cancer cells among all live cells, it stays reasonably close to its initial value. So they were seeded at 50% of either population, and so that stays more or less the same across the 3 days of duration of this experiment. And then we also have one scenario where they supplied glutamine to the coculture, and that results in a rapid cell growth. Eventually, you'll see a leveling off of the population, which we also did not include in the model. And there is an increase of the percentage of cancer cells, although it still stays reasonably close to the initial 50%. So just a quick summary. So we have done this experiment and modeling study of these cancer cells, cancer-associated fibroblasts, and their coculture. And with some exceptions, and I'll return to one of them, we have a satisfying fit of the model to the data. Of course, this is always in the eyes of the beholder, or in the case of papers, in the eye of the reviewer. And I'll come back to this in a moment. So before we go that, we tried to find parameter values in the literature, and that's actually quite a challenging job. There aren't really that many comparable modeling studies, but what we found were some experiments in which they investigated the growth behavior of various breast cancer cell lines when supplied with glutamine. And here are some results from a paper that's almost 20, well, it's over, yeah, it's almost 25 years old now. So some cell lines, they they stay constant at relatively low glutamine supply, and then they grow at comparable values of glutamine over comparable period of time. But however, this can also depend on the choice of the cell line So there are, if you only look at the doubling times of the cancer cells, they are between 20 and 75 hours, which is somewhat similar to ours. And then in another paper, which is more recent, we found that the glutamine is consumed from, it goes from 2 millimolar to 0 over approximately 3 days. And that also for various types of cancer cells. So in a way, that matches what we have seen. What was that? Here. Though not exactly. I mean, the glutamine goes down, although not exactly to 0, but it, you know, in a comparable fashion. And again, I mean, that also depends on the cell line, and ours was not among those that has been studied already previously. So, and then we had a reviewer who told us, you cannot get my stamp of approval if you keep this figure in there, right? So this is the declining cell number in the coculture. So we could claim that we have a fit up to 48 hours, but then it drops drastically, and that's not reflected in our model. And so that sent us back to the drawing board. And we decided to introduce a crowding effect or a stress factor, which somehow is produced by both the cancer-associated fibroblasts and the cancer cells. And it has an effect. It kills both cell types in a nonlinear fashion. So we had to do something. We had to expand the model. And so we added another differential equation, which is this stress factor, which we denote by L. And it's produced in, so you can say, in collaboration by both the CAFs and the cancer cells. And then its effect on the cell populations is that it is another loss term, which we decided to make quadratic in L. So it's convex. So at low concentration, it doesn't have much of an effect, but at higher concentration, it does. And concentration is a, you know, it's itself a choice of words. So we don't really know what it is. But we're having then these two additional parameter values. And that results in a better fit of the cell numbers in the coculture scenario. And that's perhaps also an answer to the question that Daniel asked earlier, right? So at what point does it become sensible to increase the complexity of the model in order to achieve a better fit? And so here we thought we could justify the increase of complexity. So you get 2 extra parameters. That's not too much. And you have a much better fit of the cell number. Yeah, so part of what we are hoping for is that eventually there will be curated databases of parameters, mathematical modeling parameters for various cell types that are commonly used in cancer research. There are already such databases, for example, this European collection of authenticated cell cultures, where, for example, one can look up the entire genome of such cell lines that are used in cancer research. But at the moment, there are no, to the best of my knowledge, ways where to look up such growth rates or death rates or metabolic parameters. So this is, in my opinion, still missing. And yeah, so the original motivation of Emily to do this kind of experiment was to see if eventually perhaps the reprogramming of the calves can be reverted. So they are the normal fibroblasts are turned into calves in the presence of the cancer cells. And is it possible to somehow revert that process and to normalize the calves and thereby to normalize the tumor microenvironment? And so they would no longer give this support to the cancer cells. And there are papers coming out that propose normalization strategies of the cancer microenvironment to improve cancer therapy. So in this case, cancer immunotherapy. So the idea is that by making the cancer microenvironment more normal, closer to a healthy tissue, one can deprive the cancer cells of their destructive potential. And it's very common to see nowadays that mathematical models are an integral part of this type of research. And I think that will only become even more prominent. Of course, I'm talking to a mathematical oncology institute, right? So I don't need to advertise this here. So then I want to spend just one more slide on what it is that I'm currently working on. And that concerns the disruption of the glucose insulin metabolism of the healthy body in which a cancer is present. So this also grew out of conversations with Emily. So healthy cells, in other words, the entire rest of the body, is competing with the tumor for glucose. And the tumor has an ability to disrupt the glucose uptake of the healthy cells, which is known as insulin resistance. And that's a condition that's akin to type 2 diabetes. And yeah, so I already came up with a mathematical model and did some numerical simulations and asked me if you're interested. There is an idea that this disruption could also be prevented by using drugs which are commonly used to treat diabetes. So for example, metformin, and that would prevent the loss of body mass. So many types of cancer are associated with cachexia, especially in their late states, where the body actually self-destructs in order to provide glucose for the cancer. And also, it's a common observation that some types of cancers have higher incidences in diabetics than in non-diabetics. And yeah, so we would like to know which cancers are and what are their properties over others. And yeah, so this may also provide an avenue for supportive treatment, not to eradicate the cancer, but rather to improve the quality of life of the patient. Yeah, so I'd be happy to find a collaborator on that project. And yeah, with this, I'd like to thank our colleagues for their contribution to this work, and also to Simon's Foundation, which supported me traveling to San Diego once. And the paper is available for download. Yeah, thank you very much for your attention.
Thank you very much, Peter. That's a very nice presentation very interesting model and very concise as well, very clearly targeted. I mean, in terms of like everything is very well motivated. So this stage, if anyone has questions, this is a good time.
I've got a couple. So as you know, Peter, we've done a bit of work on tumor stromal interactions over the years. And there is sort of an idea that stromal associations, so cancer-associated fibroblasts, have potentially genetically been changed so that it isn't, there's no way to easily transform back, right? But perhaps if those cells die, then that's how you lose them, sort of thing. So, that this idea of being able to sort of normalize the environment is an interesting one. It's one we've thought about. But the big question there is, where's the evidence to support it? And, how does one model that if you don't really have the evidence?
Yeah, I mean, I would need to talk to Emily again. There's some, this contains a couple of references on what has been done, not how successful that has been, but yeah.
Yeah, but that seems more immune associated,
yeah.
As opposed to fibroblast driven. Fibroblasts are known to have this kind of, I don't know, dual relationship where on one hand they can be promoting and on another they can be inhibitory. And that's something, David's early work and his postdoc looked at that sort of idea in prostate cancer, right, David?
Yeah.
And then subsequently we've looked at it, when there is actually sort of this idea that stromal activation can even drive stromal activations, so that the stroma themselves can activate the stroma. But again, in some of the work that Noemi Pico did in her PhD thesis looked at this question too, which is if we were to allow activation to relax somehow, or a return to normal behavior, so they become deactivated, if you like, then that could potentially facilitate a kind of treatment strategy. But the debate, the critique we always got from the biologist is, where's the evidence for that? And so I think that we never really published that work or pursued it too far because of that. And in fact, the evidence is sort of pointing towards more, I'd say, that not only do they change, but in a genetic sense, that potentially they're also like the stromas mutating and forming kind of distinct behavioral subpopulations as a result. Because we always view kind of the mutational aspect as to be projected onto the cancer, but the implication is it might be also the stroma too. So it's a kind of interesting and open question. So I definitely would suggest it's an area you should dig a bit deeper into, and if your collaborators working in that space probably knows about this debate, but it's an interesting point.
Sandy, if you could let Audrey become a panelist, so she's got a question.
I thought I'd invited her, but maybe I hadn't. I shall do it right now.
Yes.
Hopefully I just promoted her.
Cool. So Audrey, please, I know you have a question.
Yeah, thanks. So I'm just wondering if you could highlight what you feel is the sort of takeaway biological insight that your model was able to give these experiments that maybe wouldn't have been achievable by experiment alone.
Yeah, okay, that's a good question. I mean, in a way, I do think that well, I'm a mathematician, so mathematical modeling is necessary for every kind of experiment, right? So that's already my take. But I do think that well, you're good when a reviewer sends you back with questions and sends you back with homework, and so we had to introduce, for example, this loss term. And it's mysterious, and I admit we don't have a way to pin it down, but the model without would not have been able to explain this behavior.
Yeah.
And so the coculture is more than to put the two cell types together and to let them do their own thing, but in the same petri dish, right? So that I would say is an insight of mathematical modeling.
Okay, thanks.
Cool. Any more questions?
If there's no others, I have one more follow-up.
Oh, please go, but when we go for Heiko, after you.
Oh, sorry. I was wondering if there was, at any point in the experiments, any attempt to remove any of the metabolites to see how the growth would be affected. So if you removed the ammonium, for example, would it keep growing, or would you still see this crash?
Well, the simple answer is no. I mean, I have to say those experiments were done when I wasn't even there. I mean, I was giving this in form of Excel spreadsheets, and then see what you can do. This also, I have to say, it's surprisingly difficult to have people in a lab do experiment because you are suggesting them as a mathematician. And I've learned that they have their grants, they have their all their classical cancer research. I was really happy to get this kind of data that we could make a paper out of, but it's something that was done on a weekend because the postdoc really felt like doing this as well.
Okay, all right, thanks.
So I know Heiko's next. Sandy, can you make Heiko panelist? For some reason, he's joined as a guest. I'm not surprised Heiko has questions. He's very keen on this kind of approaches, so he'll probably have questions/suggestions.
Hey Peter, good to see you. Thank you so much.
Hey.
So you comment quite a bit on your parameters and suggested that some may be more sensitive than others. Are those uniquely identifiable? Were you able to maybe? I know you weren't able to fit the model, so you added different parameters. Were you able to take some away that?
Oh yeah, no, we didn't do something systematic. I mean, we didn't do some set something to 0 and see how much worse it gets and things like that. But this is the bootstrapping. So where you just add some random noise to the model output and then refit it and see how that changes and how that drives the parameters to another value. I mean, there's also this whole issue of multiple local minima, which we also didn't explore. So so I mean, there is a bit of a pattern that some of these have relatively narrow funnels. So the K1 and K1 here, and the R1 here, and the R1 here. So if we had to make a suggestion what to study in the future, it would be those.
Very good. Thanks so much.
Cool.
Hübsche Fliege.
Danke.
All right. Thank you very much, Peter. This was a great presentation. Yes. Look forward to you.
Last question.
Oh, okay.
Sorry. So obviously, the interactions between stroma and tumor cells is a spatially, physically spatial event, but you chose to sort of ignore that aspect. Do you think you're justified in ignoring that?
Yeah, I would say so. I mean, those were experiments in a petri dish, and I assume, planted the cells in well mixed fashion. I have no information that it was otherwise. And that's the.
That's for the experimental.
That's the data.
It makes sense, right?
So what drives my thought, right? The data that I have are just time points, right? And so the most natural thing is to start with an ordinary differential equation model and see if we are lucky with that. And so I think that we were. If there was anything that would suggest that there are spatial effects, then we would also need spatial observations.
Yeah. I mean, I guess that it makes sense for the way the experiments were done because you're basically activating the fibroblasts, right? But if that activation was actually driven as a result of interactions between a tumor and the fibroblasts, then there could be some spatial aspects there, right?
Yes.
Yeah. Okay. Thanks.
Right. Once again, thank you, Peter.
Yeah. Thanks, Peter.
Thanks for having me.
Bye.
Okay.
Bye.
Bye-bye.